Hello dear students. So this is our second video lecture of chapter 5, Principles of Inheritance and Variations. As we have studied, Gregor John Mendel is known as father of genetics, and he has talked about different traits found in garden pea. So there were total 7 contrasting traits that has been shown through law of dominance and law of segregation in case of monohybrid cross. Monohybrid cross means when only single gene inheritance has been shown. So as in the previous lecture, we have talked about that how the parents, once the gametes are formed and then they are going to form F1 generation. After F1 generation, they will self-crossed with each other, and F2 generation will form where all of them will not be tall, but 25% population will be pure tall, 50% will be hybrid tall, and rest of the 25% will be dwarf characteristics. So let's talk about now the various factors that affect law of dominance. Number 1, according to Gregor John Mendel, he said that the characters which are controlled by some units, discrete units means which are present on the chromosomes, these are known as factors. And these factors, they are not solitary, but they are always appearing in pairs. In a dissimilar pair of factors, like if it is capital T and small t, then it will appear as tall but not as dwarf. So means capital T is representing dominance and the other small t is showing the recessiveness. So law of dominance is very useful in identification of the parental characters, but only in case of monohybrid crosses. So definition could be this one also you can learn. In a cross of parents that are pure for contrasting traits, only one form will appear. One form means which is superior, which is dominant. Now coming to second law of segregation, where when F1 generation means hybrid tall plants are self-crossed or self-pollinated with each other, then we will get a result differently, means genes will get segregated. So during the formation of the gametes, 2 alleles which are responsible for any trait, they will separate from each other. So simply segregation term means separation. So this kind of law, it has shown us that alleles will not show any mixture. Blending means a mixture. So we cannot say that if there is one red colored flower and second plant with white colored flowers, so it will show during law of segregation that the F1 generation plant will be a mixture of red and white. No. When we are showing law of dominance and law of segregation, we are always talking about dominant and recessive characters. Means one should be able to suppress the effect of the others. So either it will be red colored flowers in F1 generation if red color is dominant, or white colored flowers will appear in F1 generation if white is dominant. So this you have to remember. According to Mendel, in law of segregation also there is no mixture found. So both the characters, they will be recovered back. Why so? Because when we have talked about that pure tall are crossed with pure dwarf, what was the outcome in F1 generation? It was hybrid tall. But when these hybrid tall were crossed with each other, when they were self-crossed with each other, then the outcome was that the parents also appeared in the same case. It means pure tall was also found in F2 generation, hybrid tall was also found in F2 generation, and even with the dwarf characteristics. Right? That was not appearing during F1 generation. There were no dwarf plants appearing. There were no pure tall plants were appearing. So parents contain 2 alleles, but during the gamete formation they separate out and the next generation will receive any of the factor. So homozygous parents, they will produce which will be similar. Like if I say capital T, which kind of gametes will be produced? Only capital T. How can they vary from each other? If there is small t parent, then again gametes are same. But if the parent is only capital T, small t, Now which kind of gametes are produced? One is capital T and other is small t. So when a new kind of trait is formed from the parents, we call it as either hybridization or recombination. So it is a result during the segregation effect. Okay. So according to Gregor John Mendel, during monohybrid cross, genes either they can show a dominance or they can segregate during gamete formation. Now let's come to the next kind of law. This is not proposed by Mendel, but the other colleagues and other scientists who followed him. So this kind of incomplete dominance, it neither show law of dominance nor law of segregation. Means when during the parent's condition, one red colored flower plant was taken and it was crossed with white colored flowers. F1 generation came to be pink in color, neither red nor white. So means we can say none of the traits are dominant over the other. So this is called as incomplete dominance. So experiments on peas were repeated by using different, because there were 7 contrasting traits. It was not only performed on the height of the plant, it was performed on the shape of the seeds, color of the seeds, then flower position, flower color, pod shape and pod color. But some of the results found that it was not resembling of any of the parent. Like here F1 generation, you can see it is pink in color. Do it resemble to any of the parent? No, but it is a combination. So it is seen in the very most common flower. You all have seen this flowers around in the garden also. It is snapdragon. So snapdragon flowers, they are appearing in 2 colors. One are red and other are white in color. It is known as dog flower. And scientific name is Antirinum. So it is a very good example to understand incomplete dominance. So how we are going to start with this, that whenever we are going to take up 2 parents, that number 1 is, it is capital R, and the second one is taken as small r as white. So the gametes formed will be capital R and other will be small r. So what we will get in F1 generation, that is a combination of 2, capital R and small r. So the flower color will be pink. It will be neither red nor white. So what will be the phenotypic ratio? At the F2 generation, phenotypic ratio is 1:2:1, means 1 flowers which are red in color, 2 that is a combination that is pink in color, and the other third one it is white in color. So genotype ratios, they were exactly similar as a Mendelian cross, but phenotype ratios they vary from 3:1 to 1:2:1. So it means we cannot say that capital R is showing any dominance over small r. Otherwise, all the flowers that appear in F1 generation, they should be red in color. But now our case is that they are pink in color. So there is a combination that came over here. So this is a law of incomplete dominance, where neither of any of genes is not showing any dominance. So genotypic ratios and phenotypic ratios, they are equal, but according to Mendel, he has shown that the ratios which are phenotypic, they are always 3:1. So this is the deviation from the concept of the law of dominance. So in most of the diploid organisms which have such kind of traits, so they can consist of 2 copies of each gene, that means pair of alleles, and they are showing either the concept of dominance or incomplete dominance. So this cross is very important. You have to remember the type of gametes and the kind of the parents which are present over there. So if we take one another example of a gene that mostly contain the information, because whenever genes are there, they have to produce one or another kind of enzymes, and enzymes are always proteinaceous in nature.
So in this case, when enzymes will get synthesized, they will have again two copies of genes and even two allelic forms, which are responsible for showing the combination of the result of both of the parents. They will neither resemble to one of the parent or the other. So this combined result is called as incomplete dominance. So from this we are getting instances that everywhere we can't apply law of dominance. So in case of enzyme, theoretically, if the allele will be modified, what it will be responsible for? Either it can show a normal or less efficient enzyme, non-functional enzyme could also be there, or it will never produce any enzyme activity. So if there is any change taking place at the genetic level, the enzyme will also change. So this is again a result of the functioning of the allele will also suffer from it. So till now we have studied about the Mendel's law, that is law of dominance and law of segregation, and this law is deviation from Mendel, where we have studied that there can be the incomplete dominance. So modified allele that is formed, it is equivalent to unmodified allele. It may produce the same phenotype or it may change the results. So such kind of results, they are very common, but if there can be chances that the modified gene is producing either non-functional enzyme or no enzyme. It means the whole outer appearance will get affected. So phenotype will only be dependent on the functioning of the unmodified allele. So which represents the original genotype. So here it is very important, if any change happened, for example that they have said in recessive trait, it can be due to non-functional enzyme or it can be that no enzyme is produced. So if a gene or allele is changing, it is resulting into no enzyme or less efficient enzyme. Next topic, what do we have? The number fourth one, that is called as codominance. Codominance neither resemble dominance law nor incomplete dominance. It having a very common example, that is human blood group. Generally, if I ask you a question, how many blood groups in human do we have? So answer is 4. They are A, B, O and AB. But how many alleles are there that are resulting into 4 kind of blood groups? So how many alleles are there? So in our red blood cells, the plasma membrane means the covering of the RBCs, they have sugar polymers which are coming out. They're acting as a receptor and the kind of sugar present in them is controlled by the genes. So gene, what is the name of the gene? That is capital I. But how many alleles are there? They are 3. It can be capital IA, capital IB or small I. Now what is the meaning of codominance? If capital IA, capital IA is the combination of the genotype, what will be the blood group? It will be A blood group. Number second, if there is a combination of capital IB, capital IB, the blood group will be B. But if they both are combined with each other, they are neither showing A blood group nor showing B blood group, but they are showing a different blood group, that is AB. So it is telling us that for the 4 kind of blood types, that is A, B, AB and O, we could have different genotypes. It is a result of codominance. Small I, if it is present with capital IA. So which one is dominant? Small I or capital IA? Obviously capital IA. Blood group will be A, and if small I is present with capital IB, the blood group will be B. But if both small I, which are the recessive alleles, if they are present together, then they are showing O blood group. So this you have to remember. Allele capital IA, which antigen it produce? A. Capital IB allele produce antigen B. But small I, because it is recessive in nature, it does not produce any kind of antigens. So for the different kind of genotypes, we have total 6 combination of genotypes, but phenotypes are only 4 in number. Phenotypes means blood group, genotypes means genetic constitution. So sugars which are present, they are expressing themselves because if capital IA and capital IB are coming together, so co means together, then they are resulting their own type of sugar which is called as codominance. RBCs, both A and B type of sugars, they can be produced. So there are 3 different kind of alleles. I am repeating: capital IA, capital IB and small I. Their combination will result into different kind of blood groups. So how many total genotypes are there? They are 6. But how many blood types are there? They are only 4 in number. That's why it can happen that one of the parent is having A blood group and the other is having B blood group, and there are chances that the child could have either A blood group, B blood group or AB blood group. So this is neither a result of dominance nor incomplete dominance, but this is called as a special law called as codominance. It is also known as multiple allelism. Note down this codominance, this showing of the blood group in humans, because we have 4 different kind of blood groups which are controlled by only 3 alleles. So this is called as multiple allelism because there are different alleles which are controlling the blood groups. Now the very important topic is that which kind of antigens and antibodies are present in our blood groups. If we usually say the person who is having O blood group, he is a universal donor, and the person who is having blood group AB, he is called as a universal recipient. So why there are chances that the person with blood group O, he can be a universal donor, means he can donate his blood to any of the other blood group person, and the person with blood group AB, he can be a universal recipient, means how his blood is able to receive any kind of blood, means of any blood group. So this is resulting due to the presence of different kind of antigens and antibodies. So let's study the table 5.2, that is table showing the genetic basis of blood groups, which is responsible for O and AB blood group, acting them as universal donor and universal recipients respectively. In this case, if we talk about the different blood groups, it means these genotypes and phenotypic nature of the genes, they are responsible for different kind of blood groups. Like if small I is there, it is coming along with the dominant capital IB. It is showing B blood group, but when small I is combining with small I, neither of the gene is dominant, so they are showing a separate blood group, that is O in nature. But when small I comes with capital IA, then they are showing A blood group, and when both capital IA and capital IA comes together, they are showing A blood group. It means capital IA and capital IB are dominant over small I, but when both of them dominant ones come together, they show a combined effect. There is no in between. They are showing a combined effect, means neither A blood group nor B blood group, but they are showing a new blood group, that is AB blood group. So this is very important. The blood groups are important whenever the transmissions takes place during the transplantation of different organs from donor to the recipient. It is very important, first of all, to check the blood group of the person. Otherwise, there could be rejection of the organs from the recipient. So in this you can see they have shown allele from parent A and from parent B. Either you can call it as parent 1 or parent 2. They have shown different combinations of genotype and which kind of blood types they could have. So if we consider blood group A, so you can see they have antibodies B. So always opposite. But blood group B is having antibodies A. Blood group AB does not contain any kind of antibodies, but blood group O has both A and B antibodies. Now, if we talk about antigens, blood group A has A antigen, B has B antigen, and blood group AB has A and B antigens, but blood group O, it does not contain any kind of antigen. I hope this must be clear now that when we talk about blood group O, we call it as universal donor because it does not contain any of the antigens. But when we say about blood group AB, we call it as universal recipient because they do not contain any kind of antibodies. So it can receive the blood from blood group A person, B person, AB or O. Antibodies are those proteinaceous chemicals which are responsible for providing immunity in the body. Thus, if any other blood group is coming, there will be no rejection by the blood cells. So, it will allow each and every kind of blood to enter in the body. Next example, as you can see in the figure of this codominance is that there can be animals, the one where you can see they are white skin color, other is red, but in the F1 generation there can be a combination. It is not that they have shown different color, but patches of red and white could be seen in the F1 generation. So this is called as codominance, which plays very important role in different characteristics. So as already I have said about ABO blood grouping system, that it is also an example of multiple alleles. So there are more than 2 alleles. So they are 3 in number. So but they are governing the same character. What is the character? Blood group. So since one individual has only 2 alleles can be mostly present, but when populations are studied, we have found that there can be multiple alleles also. Let's study one more concept. So we usually say a single gene will produce only single effect. There is one example that in pea seeds, one gene is there that is producing starch. So starch is very important because whenever food is synthesized, it is always stored in the form of starch. It has 2 alleles. One is capital B and other is small b. So starch is synthesized effectively by homozygous genes. So it means a grain